Thank you so much, sir, and good morning, all. And thank you so much, Prashant, sir, for inviting me to deliver a talk on agriculture bioinformatics. So I would like to present my work here. Hope you are able to see my PPT. Can you please confirm? Okay. So my topic of talk is contribution of open source bioinformatics platform in crop improvement. The primary goal of that talk is to brief about the how we can utilize the open source platform like Ensemble plants and other genomic platform for the identification of candidate genes and as well as gene and transcription factor families in different crop plants that are important for the purpose of food security. So first, we know about all are almost aware about the bioinformatics. So bioinformatics is to extract, store, and to analyze the biological data, and you can contribute in this field as a biologist. If you are working in lab and you are a molecular biologist, you can do work on DNA and protein sequences. You can clone them, and you can go for the analysis. And as a computer scientist, you can also develop some tools and algorithms. So this is they frequently help in analyzing the data. And bioinformatician, we study the biological questions to with the help of analyzing these molecular data. And these are the different fields are there. So these fields can be targeted by molecules, DNA, RNA, protein. It's all upon depend upon your interest, what are your interests and what would you like to do. So flow biological databases, they are the key actually in bioinformatics. So as a molecular biologist, you will do some work in lab and then you will go for the deposit this in database, and then you will go for the bioinformatics to analyze this data. And there are three key databases like EMBL, GenBank USA, and DDBJ also, DNA Data Bank of Japan. So they are the mutually collaborated to each other on daily basis and share the data. So using these databases, you can access the lot of data from there that is free of cost. So now the key resources for big data, as earlier I told, that is NCBI, EBI, and DDBJ. There are big data for the uh, analysis of genomic data, proteomics data, metabolomics data. So everything is there. There are some online tools and some standalone tools also available there. Some, if you are familiar with the command line, then you can also go for some commands there and you can analyze your data, retry your data, and store your data in different formats. So that is a facility basically for these databases. And different omic levels, you may aware about the different omic level in bioinformatics. So this is phenome, static phenome, metagenome, microbiome, and metabolome, functionome, proteome, epigenome, transcriptome, genome. So we people are working on different aspects of omics. So this is our target areas, and we are working on these fields since 2013 at Prashant told. So in different collaboration with different organizations, Prashant is also including this as a collaborator and as my mentor. So now we have the multi-omics for crop improvement. So how we can utilize multi-omics in crop improvement? So the flow of omics level starting with the genome, the genomics, transcriptomics, proteomics, and metabolomes. So we can go for the, if you are working on genomics, then we can go for some analysis like antennae, and we can identify some key genes. In transcriptome, we can go for the whole genome alignment to find out the variations, and proteome we can go for the protein structure modeling, and metabolome we can go for the study of pathogen, how they affect the pathway and what are their key pathway and how they are involved. And the observation at phenotype level to facilitate us to conduct some analysis like GWAS for the and QTN analysis to find out the relation between genotyping and phenotyping data, as well as to find out the key genes that are upregulated and downregulated and play an important role in response to a plethora of environmental stresses, including a biotic, and like and others is about the protein interaction network. We can go for the identification of how they are interacted to each other at the level of proteome, And we can also go for some docking studies, and docking studies facilitate us to find out the interacting residue, how they are interacted to the protein, and how they affect the overall function of the particular gene or a group of gene. So here the popular plant genomic resources that we are frequently utilizing, and among them ensemble plant is the one of the most important and my best. So actually we are utilizing ensemble plant since 2015, and so far we have published more than 30 paper with utilizing ensemble plants actually. So this plants is, and yesterday ensemble plants turns to 22 years, and they celebrate 22 years of establishment of ensemble plants, and that is providing access to free of access to so many crop plants and their genomic data. So we can, if we are interested in any crop plants, we can directly go there and we can download the data from there. There are so many tools for the data mining like Biomart, Blast for the homology search and SNP detections and much more. So you can explore ensemble plants and you can find out the so many things there. And with the utilizing this ensemble plant, we have identified different gene family in not only wheat but also in sorghum and rice and other crops as well. So these, that is very important for us and can be utilized for the identification of different candidate gene also. So RAPDP is dedicated to rice annotated project. If you are interested in rice, then you can utilize this RAPDP. And maize GB dedicated to provide information about the maize genetics and genomics. So you can access this for the identification of some genes, and you can download data for the prediction of some molecular markers, other things, and QTL mapping as well. And then TAR is dedicated to model plant, the Arabidopsis information resource that provide a lot of data about the Arabidopsis. So their genomic pathways, KGG, and gene ontology terms, and genomic information, proteomics information, metabolomics. So everything is there for TAR. And ARGI is dedicated to provide information about the wheat genome. So that have recently sequenced. So there are a lot of data about the wheat genomics is available on ARGI, and that is free of cost with some tools there, including ARGI wheat on wheat genome, including that is Blast and Synteny Viewer, SNP for the SNP mining, and other things are also available there. And Grammy is dedicated to provide the information about the all the members of Grammy family, Poesy family. So that is also one of the most open access platform for identification of genes and gene family. So now the application, how we can utilize bioinformatics for crop improvement and what are the major applications. So in plant breeding, if as a plant breeder, so we can utilize for the we can go for the development of some molecular marker and can utilize them in marker assisted selection, improve nutritional quality, and we can identify some key genes associated with the traits like in biofortification and their utilization, fruitful utilization for development of nutrition efficient cultivars and development of stress tolerant varieties. So transgenic approach within the utilization of genomic information. If we know what is the candidate gene and what is the expression used based on the RNA-seq data available in public repository or microarray data, then we can go for the key gene identification, can utilize for the transgenic approach. So now the potential applications. So these are the basically potential applications, and we are working on these applications since last 4 to 5 years. So these are functional annotation of annotated plant genomes like wheat, and identification of key genes associated with traits, different traits, and development of gene-specific molecular marker like SSR marker and ILS marker, and as well as microRNA SSR marker. So recently we have published 2 to 3 studies on microRNA SSR as well, and identification function annotation of transcription factors responsive to abiotic and biotic stresses in plants, discovery of microRNA microRNAs, which I have told, and protein-protein interaction network to find out the key residues, and development of biological data to facilitate the molecular biologists to access the data and utilize this for further improvement and annotation purpose. Now here I would like to present some of our studies that have already been published in different journals. So we have actually so many studies I am involved, but we have present only 2 to 3 studies. So these are the actually our case studies. So SPX gene family we have identified in wheat, and that have been published in International Journal of Biological Macromolecules. So role of SPX gene family in plants. So they are the ubiquitous in vascular plants and play an important role in development, growth as well as in response to phosphorus discharge. That is more important for the growth and development of a particular plant, and potentially function as new group of phosphorus transporters. And they are explicitly having the conserved domains that is very important for their functional stability, and that is approximately 165 amino acid long. So these that have been classified in different categories, subfamilies, that is SPX, EX, SPX MFS, and SPX ring. So well known for their important role during the iron deficiency response as well as disease resistance, and comprehensively studied in different plants, Arabidopsis thaliana and soybean, common beans, rice, and brassica. But that point of time that have not been identified in wheat. So that's why we study this gene family in wheat. So we have identified SPX gene family with the utilizing of IWGXC data, that is International Wheat Genome Sequencing Consortium reference sequence data, available at Ensemble Plants for 2.0 version. And using this platform, we have identified about 46 different SPX proteins, and all different 46 have all been characterized in different subfamilies, including SPX with 15 members, SPX EX with 12 members, SPX MFS with 12 members also, and ring with the 7 members. So distribution of TaSPX genes. So this is the chromosomal distribution of SPX gene on wheat chromosome, because wheat have three subgenomes. So based on this representation, we can say that 7A, 7B, and 7D found the hotspot for these SPX genes. So they are scattered on the SCA and SB and unevenly on SSG. The chromosomal representation based on the data and values that we have derived from in the form of genomic coordinates from ensemble plants. And then structure of TaSPX genes that have been published, that have been predicted using GSDS server, gene structure display server, and Mega 2. So based on this, as we have earlier told they have 4 to 5 classes, basically 4 classes. So including SPS, MFS, ring, and EX. So based on this, they having actually 4 classes, and these classes are associated with different domains and as well as different gene structure patterns. So here, the pink color represents the CDS, while this line indicate the introns. So they are the good combination of these genes, and they are well stable for the purpose of structure annotation. Now the phylogeny of SPX protein we have conducted using Mega tools. So this for that purpose we have downloaded the Arabidopsis thaliana SPX genes, brassica, wheat, and rice from ensemble plants. So based on this, we have conducted this phylogenetic tree, and based on this phylogenetic tree, we can conclude that there is the consent of the previous studies that have been published for the Arabidopsis thaliana and brassica. Now we can say they are the closely associated with the rice, because rice are the monocot, and they are the closely associated with each other based on the common ancestor. So they are the gene family and these gene members showing the close relationship with the rice SPX as compared to brassica or Arabidopsis, because they belongs to dicot. Now the cis-regulatory elements, we have found a lot of cis-regulatory elements in SPX. So they belongs to and they respond to different developmental and as well as the abiotic stress responses, and they are well known for their important role in plants. And this is we have identified using plant and tool for the purpose of identifying cis-regulatory elements. So we have also find out the PI1BBS. So PI1BBS, one of the most important cis-regulatory element response to phosphorus starvation in plants. So that is the key actually cis-regulatory element which we had identified. And others also. There are also include some MYB and MYC and DRE, and these are the basically cis-regulatory elements. They are frequently distributed in the different transcription factor families responsive to abiotic stress in plants. So we have also studied these SPX based on the QRT-PCR basically, and we have conducted the expression profiling with the help of South Campus actually Delhi University. So based on the expression profiling, we can say that these representative SPX candidates. So the very good expression profiling, and they sometimes they upregulated, sometimes they down. So these are the important for the phosphorus stress response in plants, and especially in wheat. So we have also conducted the molecular dynamic simulation to study the multi-omics level, their stability, how they are stable at the level of protein. So based on this we have conducted the molecular dynamic simulation, and for the graph you can say that these are in the early stage they are they are highly fluctuated and they are not stable, but after 6000 ps they are stable. So now we can say that these are the stable proteins and can be utilized for the purpose of some transgenic approach, and as well as some other like the CRISPR-Cas9 or other reverse genetic approach. So these are the superimposed structure of the basically all these is combination of proteins, and if here the different color represent the different key proteins, and here the residue fluctuation. So we can find out that before 80 they are the highly fluctuated residues. So these are residues found in the SPX 18. So there is some fluctuation, but after that they are stable. So now we can say that could be utilized for further iteration. So now the second case studies, that is about the identification function annotation of candidate genes associated with nitrogen, phosphorus, use efficiency in wheat. So that have also been published in the same journal, International of Biological Macromolecules. So, and for that purpose, we have utilized ensemble plants for the identification of these key genes. And now the importance, we know how nitrogen, phosphorus are important for the plant growth and development. So these are basically the two most essential macronutrients and for the plant's stable growth. And the component of important molecules is like nucleic acid, phospholipids, and ATP. Now here the utilized the pipeline basically which we have utilized, and the key tools and web server. All these are the open source and freely available for the scientific community. So in phase first, we have identified the genes using different tools like BLAST, gene structure, functional domain, physicochemical properties, cellular localization. Phase second, we have annotated the genes using phylogeny, molecular marker, microRNA target, gene expression profiling, interaction network. In phase third, we have conducted the molecular modeling. So based on the modeling, we have identified some homolog template for the protein structure prediction and structural alignment to find out the similarity between two protein structures, function prediction based on the profunc and the gene ontologies, molecular dynamics for their stability evaluation. So now the pipeline, so basically that is the pipeline which we have utilized. So first we have derived the nitrogen use efficiency and phosphorus use efficiency proteins from Arabidopsis, Brachypodium, and rice. So why Arabidopsis and Brachypodium and rice here? Because we consider Arabidopsis as model plant and Brachypodium rice are the monoclot plants and they closely associated with the wheat. So that's why we have selected them as a query. And then we conducted the BLAST P against ensemble plants and IWGSC ARGI database that hosts the Triticum STM database. And based on the gene IDs for best state, so we have selected the some key genes. They are having the identity 70 to 90, 33% with lower E value 0 to 1, and extracted the genomic sequences from ensemble plant for full length transcripts to predict the gene structure and other annotation as well, like microRNA SSR development SSRs. So, and then the prediction of gene structures using CDS, and then the structure annotations. So based on this study, we have basically identified 37 candidate genes associated with nitrogen and phosphorus use efficiencies. And out of 37, 21 were found associated with NUE, while 31 were involved in inorganic phosphorus transporters. and they have been further characterized in different categories like amino acid biosynthesis, transcription factors, nitrate and ammonium transporters, and phosphorus transporters. So we have also predicted some gene-specific marker and a microRNA target for these key identified genes. So these are the 23, as many as 23 SSRs we have predicted. So some are the trinucleotide, tetranucleotide, dinucleotides, pentanucleotide, hexanucleotide, and further we have validated them in wet lab actually to on different genotype to screen the diversity in a study. And basically we also identify some 10 microRNA. Basically they frequently target these candidate genes and also predict their interaction network to find out how they are interacted to each other and what are their concept, basically to study their mechanism. So this is basically gene expression profiling which we have conducted based on the based on the microarray and RNA-seq data available in Gene Investigator platform. That is also one of the most popular platform for the scientific community to conduct the gene expression profiling, not only for plant but also for the animals. So we can utilize this for the purpose of expression profiling. So that's why we have conducted the in silico expression profiling, and based on this we can say that they are our identified genes show differential expression in different condition, basically in different developing stage as well as stress conditions. So based on this we have some filter out some candidate genes and validated them in RT using QRT-PCR in wet lab actually. So these are the genes basically, and we have also predicted the protein chemical interaction network to find out the interaction between the small chemical molecules and these transporters. So we have find out some interesting molecule there. So they are the glutamic acid and glutamic acid, ammonia, nitrate. So they are the key small molecules, and they inhibit the activity of these molecules, these receptor basically. And so for that purpose, we can also go for the molecular docking and followed by molecular dynamic simulation to study their interaction, how they are interacted to each other at the level of metabolism basically. So we have conducted protein structure prediction with the help of homology modeling approach. So first we have our amino acid sequence, and then we utilize the protein data bank for template identification. So this is our template, and based on this we have conducted the sequence alignment, and based on sequence alignment we find out the homology, how they're similar to each other. And then we predicted 3D models, and this 3D model actually we have predicted using Swiss modeler, and based on this we also evaluated further using Ramachandran plot. So based on the Ramachandran plot we can say that this is good and showing more than 90% residue in favorable regions. So we can consider this type of protein structure in this our study. So here the structure of model 3D structures, different 3D structure basically for different proteins. So all these product structure have been predicted using the same approach, and and further rendered in the UCSF Chimera and PyMol for their structure annotation. And we also conducted this protein structure alignment. Now the structure similarity between two proteins basically. So here the first we have selected the ammonium transporter from Arabidopsis, and this is our one which we have identified in wheat, that is EMT ammonium transporter. Here TA represent the Triticum aestivum, AT represent the Arabidopsis thalinia. So based on this super actually FATCAT tool, that is open source tool, we have conducted this superimposed structure. So now we can say that these are less than 1 angstrom, and they are very good in similarity. So then at the level of structure they are very good, and so they can be considered for the purpose of annotations. So in second study basically we have conducted the for RWPRK transcription factors in plants. So these RWPRK are very important for the purpose of nitrogen starvations. So they have a conjunct motif within NLPs, that is NIL-like proteins, and PB1 additional domain, and RKDs, which is contain only RWP RK domain. So they're using bioinformatics approach. We have identified 37 RWPRK genes. Among them, 18 belongs to NLPs, while 19 belongs to RKDs. So these are basically their domain architecture. So based on the domains, we can say that NLP is having additional PB1 domain, while RWPRKs, RKD subfamily having only RWPRKs. So these are the predicted gene structures. So here red color represent your CDS coding pattern, and the line represent the introns, while blue view are the upstream and downstream regions. So they showed the actually combination of all these, and well good for the purpose of further characterization. And we also predicted the protein-protein interaction using the gene mania tool basically. So because weak data was not there at that point of time, but for that purpose we have utilized the arboreal data. So now we can say that the different RKDs, basically these RKDs of subfamily of RWPRK belongs to different, and show some interesting interaction with the NLP8, NLP9, NLP5. That is also subfamily of RWPRK. And we also find out some interaction with the NLPs with the RKDs. So these are the two combination of their NLPs and RKDs, how they are interacted to each other and response to nitrogen starvation. Not only response to nitrogen starvation, but also for other abiotic stress like drought, heat, and cold. So structure-based function attribution, basically, first we have predicted the 3D structure, and based on the 3D structure, we have predicted their biological function based on the profunc. So based on the profunc annotation, we can say that they play an important role in different biological process like cellular process, metabolic, primary metabolic process, metabolic, and much more. And they play an important role in binding, catalytic activity, nucleic acid binding, metal ion binding, ATP binding. So these are the basically key function of these RWPRKs. So we have also find out their interaction between basically with the nitrate. So based on this, we can say that they are interacted to the different proteins. They are the our key representative like NLP7 and then RKDs. So based on this, we can say that they are interacted to 4 residue here, and LYS51. So these can be utilized, these residue and this information we can utilize for the inhibiting designing inhibiting molecules. So that can be utilized and can be validated in wet lab for the help of X-ray crystallography and NMR. So we also developed the nutrient use efficiency web resource under the mentorship of Prashant sir and Professor P.K. Gupta sir, Kavi Kishore sir. So that is hosted on Bioclues official website. Anti-web resource basically manually curated resource and provide the comprehensive information about the genes, QTLs, and their corresponding microRNAs. So this is basically dedicated to provide information about the 4 major cereal crops, including wheat, rice, maize, and barley. And 2 LN species like Ureth to Angelopis are also included in this. And in model plant, we have considered the Brachypodium and Arabidopsis thaliana. So here the pipeline which we have basically utilized for the purpose of development this anti-web resource. So this is we have mined the data from ensemble plants primary database, GenBank, Graminy, UniProt, and PSRNA target for the prediction of microRNA targets, and UniProt for the protein information, Graminy for the QTL information, GenBank for the genomic information, ensemble plant for the other all information like their physiochemical properties and their gene structures, as well as their pathway. And the information about this basically we have derived from the literature, that is about the trait information, their gene description, QTLs, protein function, microRNAs. So based on this, we have manually curated the entries and stored in MySQL and designed a web page on Java. So this is the basically home page for this, and this is provide the home advanced search and about web resource, team information, FAQs, and useful links. So that is actually active and providing information about the nutrient use efficiency associated genomic elements, including gene, QTLs, protein, and microRNA, as well as transcription factors. So here the information available in NT resource, what is available there. So first one is the plant species which we have covered here, the macronutrient about the boron, chlorine, copper, Mn, Mb, zinc, and Ne, and my macronutrients also NPK and gene annotations like the gene name, gene IDs, gene description function, and microRNA target information, target microRNA accession, species name, and information about the QTL also available there. So that provide information about the putative QTLs, their chromosomal location, and trait information reference with the literature that I will publish already in peer-reviewed journal. And it is the platform basically which we have utilized, that is MySQL and PHP. So here the home page that facilitates the end user to access the data with the basic search and also as well as advanced search. In advanced search, end user can define the particular plant in which he is interested or interested. So he can access the data and this search for result and nitrogen associated gene. This is the example basically. So based on this, you can enter here the more information, and based on this more information you can visualize the information like the organism name and type of elements basically, that is whether this is macro or micro, gene information and gene hyperlink that is associated with the reference like ensemble plant or like GenBank or EBI, and information about the literature also that have been published in journal for the QTL studies. So these are the information basically. So here the publication basically we have published so many studies there, and we have published so far more than 50 peer-reviewed article in different journals, and collaborated with the different lab like Professor P.V. Kavik Shore sir lab and Prashant sir lab, Professor P.K. Gupta lab, and ISRI, and so there are so many labs there. So I'm associated with them to provide the bioinformatics support. So any question, please? And I would like to acknowledge the our collaborator and the Ajay Pandya and other Girik also who developed this database and provide the technical support. Thank you so much. Any question, please? Hello.
May I?
Yes.
Good morning, sir. May I?
Yes. Yes. Please.
Yeah. Uh, Thank you for such a informative talk, sir. Actually, I have a question that is there any database or something like that, because I don't know to get information about the plant secondary metabolites. That if I know the plant and then I have to look for that what type of scam metabolites they are producing. So, is there
Yes.
any database?
Yeah, actually, there are so many databases you can explore. Like NPSS is there, and something or other. So I can send you in my email. Actually, right now I know only NPSS. Other are also there, so I can share with you. So you can send me your email ID. My email ID is here. You can share and ask me. Then I will share you some information about the metabolite specific databases.
Okay.
Any other, please?
Okay. And,
Yes, please.
sir, I have one more question. May I?
Yes, please. Yes, please ask.
Sir, actually, I'm doing some work in this field of this plant biology and all that. So actually, there is a problem that we know the local names of the plant. Yeah, we know the local name of the plant, but we are very much not informed about their scientific name and something like that. So is there any resource or something like you can recommend any book or something like that so that we can get the information about the binomial nomenclature and the
Yes, yes.
scientific name of the plant?
For that purpose, Yes, yes. For that purpose, what you can do,
We know the local name, so
okay, okay. So for that purpose, what you can do, you can directly go for the phylogeny platform of NCBI, and there you just put their common name of your plant, then you will be able to find out the entire taxonomy of particular plant actually. So that is for this. And Indian government also environmental department also developed the name of like just vernacular names like app is there. So I will share with you. So that app also you can utilize not only for the scientific name purpose but also for the vernacular names. And also we have also published 3 books on the basically medicinal plants. This is for
am I
medicinal
audible, sir?
plants. Yes, yes.
Yes.
So that is for the basically medicinal plants for distributed in Uttarakhand. So that have already been published, and we have provided the scientific information like their taxonomy and their metabolites in these plants. So book. So you can access this also. I will share you information. So phylogeny NCB phylogeny is there basically.
Okay. Thank you so much, sir. Thank you so much.
Just
Okay.
providing link. You can access this link actually for phylogeny purpose. I am providing link for the taxonomy of wheat actually. For example, you can use this type of. So you can type any
So
plant
thank you
there,
for the information.
and you can access the taxonomy information about particular plant there. Any other, please? Okay. Yes. Okay. Yes. Yes, actually, so best thing actually is I think is depend upon the genome data availability, and I would like to recall that when I joined the Professor P.K. Gupta lab in 2012 there, so I was unaware about the how to identify gene. I was very good in bioinformatics as theoretical purpose, but I was not very much frequently with the practical bioinformatics to identify the genes. So there I learned actually under their team guidance, Professor Cis and Professor P. Gupta, I learned how to identify basically the candidate genes and how can map their chromosomal location, not only in using bioinformatics, but also in the wet lab actually. So that was the actually that time where I learned about this. And the difficulties is basically depend upon the data and how we can understand this data basically, because there are so many data versions is there. Some are annotated data, some are unannotated data. So based on this, we can utilize, but we should have strong pipeline. We should have strong concept. We should know about this, what is the function of that gene previously in other crops. So that is the reason actually. So we should have all these not only technical skills, but also as well as some biological concept. So that is very important for the screening and for the purpose of mapping actually. Yes. Not yet. Not yet. But I will definitely make a plan for that very purpose. Thank you so much for your suggestion. I will definitely make a plan to study SPX in plant in animal as well. Yes, yes. They actually they yes they have been explored in plants as a phosphorus transporter and have been studied as the uh, five phosphorus transporter family actually they belongs to. So that is very good to study them in animal also. So I will definitely make a plan for the very same in future. Yes, 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 yes. Yes, 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 yes. There is actually PlaxDB is there. That is plant exhibition database. And second is about the yes. That is not working right now. But if you are working in wheat, then you can go for the wheat XPDP. And if you are working in sorghum, there is sorghum FG, as well as for rice also. And for to clustering these heat map and to retrieve the RNA-seq data, you can utilize the Gene Investigator platform is there actually. So you can utilize Gene Investigator platform. So I have utilized Gene Investigator platform as well as Wheat XP DB and sorghum FG for the identification of sorghum candidate gene and their expression profiling, which we have worked for the Professor Kabi Kishore sir lab actually. So that is the actually for that purpose we have utilized the sorghum FG. So they are the actually and some they are actually frequently publish some research group in the papers on the and their databases on their gene expression profiling actually. Now they have a RNA-seq data and they have counts. So they are adding there in database version. So we can access the expression profiling information from there actually. In future, I think that will be available in ensemble plants also. So I think second talk is about the ensemble plant. So we can discuss with him. Then yes, yes. Thank you so much. Thank you so much, sir, for inviting me this and for your mentorship.